Welcome back, folks. This video lecture is on Chapter 15, Translation. I've distilled the PowerPoint down to the essence, and we're going to focus in on some of the figures that are key to understanding this chapter. Where do we begin? Well, let's begin with a simple landscape picture. We need you to know that DNA is the information carrier for all cells. All life depends on DNA as far as we know. And the information in the DNA is extracted and is converted into RNA. And there's many types of RNA, as we now should be realizing. And some of the most important RNAs are going to be messenger RNA, ribosomal RNA for this chapter. Let's put it over here, ribosomal RNA. And then, of course, the third player, which is transfer RNA. And when all three of these mechanisms of RNA activity come together, we end up getting something called translation. And that is then turned into a protein. And this happens in all living things, as I just mentioned. All right. And we also learned in the previous chapter, chapter 14, that this stage is called transcription. So transcription is the conversion of information into RNA. And today we're going to look at translation. And translation is the step, the complex step, where information is converted from messenger RNA into polypeptides, and those polypeptides, once finished, they go to work and are now known as proteins. Where do we begin in a complex chapter like this? Well, the first part of the chapter has a very simple historical perspective, where we give credit to people, scientists in the 20th century who were able to observe patients or observe living organisms and then make deductions. And those deductions have been profound in our understanding of how, in many cases, biology and genetics and biochemistry are interrelated. Okay, so this is a good time to use an analogy. So the analogy is that we have a water body, a body of water like a reservoir, upstream on top of a mountain, on top of a hill, and then it has a small river that flows into a pond, and that pond has water that spills over and runs into a second pond, and from the second pond we have water that runs into a third pond, and from the third pond the water runs into the ocean. Okay, so that analogy we're going to use to understand biochemical pathways and their genetic interactions. Now, assume that in this body of water, we have locks. So let's put the locks in a different color, because they are the essence of what we're trying to learn. So let's choose a blue color. Okay, got it. Now, assume there's a lock here. And once you open the lock, you allow water to flow out of this body of water down this stream or river into this body of water. And this body of water will have its own lock, as will this one, and finally the last one. So we have lock A, lock B, lock C, and in this example, lock D. And this pathway to the ocean from this reservoir is pretty simple. It's a linear pathway. And that's what we assume that most biochemical pathways they follow this pattern, right? And on the other side here, we're going to use a different color, and we're going to relay that information in terms of biochemical pathways. So let's say we start with food entering the cell, and then that food is going to be converted into product A, and product A will be converted into product B. Likewise, product B will be converted into product C, and product C will be converted into D. And once we have D, it's then finally converted into E. And E is going to influence the phenotype of that organism. So if this is a pathway for producing pigment in a petal of a leaf, or the petal of a flower, I should say, and then the final product would be the pigment itself, that it would be produced by the plant from nutrients that it gains from the environment. The analogy to the locks is this. So each step in this pathway, we have an enzyme. So let's call that enzyme A. So if enzyme A, which is normally a protein, is produced from its gene correctly, then we will have a functional enzyme. So this is going to be gene for enzyme EA. Okay. And likewise, we have another enzyme here called enzyme B. That too comes from its own gene, which we won't draw to save time. Then we have B going to C, that's controlled by an enzyme C. And we have an enzyme D. And then finally, we have an enzyme E. Okay, and each one of these has its own gene, which has to be functioning correctly to produce the correct functional enzyme. So how does this work? Well, in this pathway here, for you to continue to supply the ocean with water, all the locks have to be open, and there should be a continuous flow of water from above to below, and then everything will be normal. If one of these locks fails, then the water will begin to accumulate at that location. So imagine that there's a failure in lock C, and then the water will simply not be able to pass beyond that point in the pathway. So as far as the ocean is concerned, there's no water flowing in. Okay, so there's a block or failure with this lock here, then everything will be fine here. Everything will be fine here, but then there will be no product D. Therefore, there will be no water entering the ocean. So if you look at it in the same context over here, you will see that if the gene for enzyme EC is defective, so that gene is defective, therefore this enzyme is not made correctly, and it doesn't function in converting B to C, even though enzyme EA is correct, and that will be working normally, and enzyme B is correct, that will be working normally, then there will be no way to convert B into C, and thus C will deprive the cell of D, and that will deprive the cell of E. And in this case, no pigment will be made. And then maybe the flower petal will be white rather than red, for instance, right? So biochemical pathways are useful in understanding how genetics and biochemistry interact. So once we know that, that's the key to this entire chapter. What we now need to learn is what is the relationship between genes, enzymes, and biochemical pathways. So we're going to do that next. So if we move to the next slide, these experiments were alternatives to studying patients. Here, fungi were used historically in the 20th century to discover biochemical pathways. So what did they do in that case? Well, let's go to the next slide on this side, and we'll try to draw that. So suppose you have a normal fungus, right? Let's put the normal fungus at the very top. So the normal fungus can take from the environment food. Let's use a different color because I need to make sure we understand this. So they use food, the fungus uses food, and then it converts that food internally into A, product A, then it converts that to B, okay, then it converts that to C, let's put a few here, and then that converts that to D. And then finally, D is converted to allow the fungus to produce something essential, right? Like a vitamin or not a vitamin, sorry, an amino acid that it needs to survive, to make its proteins, right? So it makes amino acids for use in making proteins. So you now know that this pathway is going to be essential to allow the fungus to produce the amino acids so that it can build proteins from those amino acids. And this is how those amino acids are made internally within the body of the fungus. All right. Just like in the previous slide, every step in this pathway has to have an enzyme made by its own gene, right? So there'll be an enzyme here. Let's call it A. Let's call this enzyme B, lowercase, enzyme C, and then enzyme D. And then finally, at the end of the pathway, we're going to have an enzyme E. Okay. Now let's assume that you are a normal fungus, that you have all the genes functioning normally. That means we can put you on a plate that contains just simple food. So here's the plate, and we put you on there, and we say, okay, if you can take that food and convert it through each of these steps to the amino acid, then you'll survive. And inside your body, you have everything working normally. So the genes for E, A, B, C, D, and E are working correctly. So we call that fungus wild type, right? Wild type or normal. And in terms of nutrition, it's called a prototrope, right? That means it can feed itself by making everything it needs from scratch if it needs to. Now, suppose you have a friend, another fungus, in which that fungus has a mistake in enzyme EA. Okay, so if you put that fungus, your friend's fungus, let's put it in a different color. Let's put that in green. So you have your friend fungus, and you put that on a plate like this. That fungus has a defective EA. So it doesn't have the enzyme EA, that's missing. So on this plate, it can absorb the food, but it can't convert that food to product A. So that fungus will not grow, whereas you do grow on that plate. Now, can that fungus grow on any other plate where you have supplied a different type of substance? Well, let's try it out. Suppose you have a plate. I'll stick to the same color. Suppose you have a plate here that already has product A built into it as a source of biochemistry. So it contains substance A. Can this friend of yours, this friend fungus, can it grow from this point onwards and produce the amino acid that it needs? And the answer is, well, if you give it A and the block is upstream of A, then it will have no problem converting A into B because it has a normal EB enzyme and everything else downstream. So that fungus will grow normally on a plate that's supplemented with A. A normal fungus could grow on any of these locations because it's able to grow here. It's able to grow if you give it B. So this is B being placed into the medium. This is A being placed into the medium, and this is just food. So you see, understanding this is very, very important to understanding not just this chapter, but many other chapters that focus in on biochemical pathways and their mutations, right? And likewise here, if you give it C, it will grow because it just needs to go from C, D to the amino acid, and it has all these enzymes as being normal. All right, and if you give it D, it will grow. And in fact, if you give it the amino acid that is missing, it will grow too, right? So the wild type will grow on any of these plates that the scientist has manufactured. And the mutant fungus, that will not grow on the first plate because it has a block in the pathway right here. But anything downstream of that particular fungus, we should have no problem growing upon, because everything beyond that point, everything downstream of that is going to be normal on B, on C, on D, and on plates containing the amino acid. By studying lots of fungi like this, suppose you found the following situation. So I'm going to just copy all this. I'm going to copy that and put it onto a new slide, and then paste it in, and then get rid of some of this stuff because I don't need it. Let's get rid of all that. Get rid of this too, and then get rid of that, right? And we don't want the wild type here. So suppose you came across a new fungus and you wanted to find out why it doesn't grow on a normal plate. And what you could do, let's use a different color. Let's use a bluey color, light blue. Suppose you knew that the fungus that you discovered did not grow on this plate, that plate that contains food. What you could do in the lab is get these other plates out and grow the same fungus on those plates, on a plate with B, on a plate with C, and a plate with D, and a plate with amino acid, right? And suppose you found out that the fungus grows on the amino acid. So if you put it on a plate with the amino acid already there, it's able to flourish and grow. And suppose you discover that it does the same thing on a plate that you supplement with product D. But it doesn't grow when you give it C, only C. And it doesn't grow when you give it B, and it doesn't grow when you give it A. And it doesn't grow definitely when you give it just a raw food material. So by combining these two figures, the top and the bottom, you can work out the point at which it will grow and the point at which it won't grow, and find that step. So that step points to the fact that in this fungus that you just discovered, it's more than likely that enzyme ED is mutated. The gene for ED is defective. So you've discovered a new fungus that will allow you to then relate that back to that biochemical pathway. And in the 20th century, in the early part of the 20th century, the scientists had the mutant fungi, or they used radiation to generate mutant fungi by giving them bursts of gamma radiation or X-rays. And then they were able to figure out, by using these types of plates, where the block was in that biochemical pathway. Now, biochemical pathways can be more complex. You could have a substance F joining the pathway here. And in that case, you'll need an enzyme EF, right? So in this case, if there's a block in this step here, that block could be overcome by the normal functioning of the enzyme EF. So this is like water arriving in this pond from either this pond or from that pond. As long as one of the ponds is supplying water, the water will continue to flow to the ocean downstream. So this analogy between locks and water and biochemical pathways seems to work in my head. So try it out and see if you can relate to some of the more complex examples that are out there, right? So these are the conclusions that were drawn by these scientists. They said at the time that they believed that one gene in that organism was responsible for producing one of those enzymes, E something. We now know that that was too simplified a view of how things really work. We now know that enzymes are mostly a type of protein, but proteins can do lots of things other than just be enzymes. But not all enzymes are proteins, as we'll learn in another chapter, right? A small percentage of enzymes are made of RNA. So keep that in mind. Bullet point number 2. Proteins can be composed of subunits. So if I'm a protein and I work at step EA, I don't have to be a single polypeptide. I could be 3 or 4 polypeptides working together to produce the final enzyme. So a gene could be coding for a part of an enzyme and not the entire enzyme, right? So now we say that a gene makes a polypeptide. And then the polypeptide may be used by itself or in combination with other polypeptides. Right? And at that point, we use the word subunit, protein subunit. The situation is complicated further by the presence of alternative splicing, which we discussed in the previous chapter, chapter 14. And that gives us alternative polypeptides from the same gene. especially in eukaryotes. So you see how your understanding has to be modified based on the data, but have your cornerstone, have your foundation solid, and then you can build new ideas on top of that as more data is revealed to you, right? And the final number 4, says many genes encode functional RNAs. That means not all genes produce polypeptides. Many genes, more and more in fact, they do not have translation. They stop after transcription. So that picture must be solid in your head too. So if we go back to this side, how does that fit in here? Well, that means these steps are very, very common, like this, leading to the production of many different types of RNA, including ribosomal RNA and tree RNA. And as we'll see in later chapters, at least another 45 different types of RNA are produced by a typical eukaryotic cell, right? So only a small subset of those RNAs are converted into protein. So that's the new understanding that we have. So we live in the golden age of understanding, and understanding how everything fits together in the real world is what we're after, and that's very important. So moving a bit faster now, the genetic code is how information stored in nucleic acids, RNA and DNA, in the form of nucleic acids, how that information, which is stored as bases, yes, so you're going to have A's, C's, G's, and either T's or U's, depending on if it's RNA or DNA, how that information is then converted into information that's stored in the correct amino acids. AA stands for amino acids in my world, and there's at least 20 different types of amino acids, right? And they then go on to make up the polypeptides, and the polypeptides will then go on and perform the task of generating proteins that will then change the phenotype of the cell, okay? So that's the way that life works. So we're going to look at this step here, how information stored in nucleic acids is then faithfully translated into information stored in amino acids. So that step there is called the genetic code, right? So it's like a spy trying to translate a secret message into an understandable product. Research, as I'll show you in the next few slides, has come up with a historical understanding of how we were able to deduce this secret code that nature uses. Now we know that the code consists of words, right? And the words in the nucleic acids are, in almost every case, there's always an exception, but we don't look at those exceptions. They're going to be 3 letters each, right? So A will be one word, and AAA would mean something here, and then you'll bring in one of these amino acids that corresponds to AAA. Change the word to GAA, and then of course that will mean something else to this machine that brings in amino acids, and now it'll bring in a different amino acid. So that's what we're looking at. The word anticodon only applies to the structure of transfer RNA. So let's put all this together in a form that makes sense. Here is the standard, there was the standard, here is the standard genetic table, the genetic code. Most creatures, not all unfortunately, but as far as we're concerned, 99.9% of all living things on this planet, they use this code in their nuclear DNA. We do have mitochondria, and they are slightly different for historical reasons, but there's a large overlap. So most of the code is the same. 1 or 2 words mean something different in mitochondria compared to nuclear DNA. There's a few protozoa, single-celled organisms that use a slightly modified genetic code. Why? We're not sure. It could be due to evolutionary pressure. All right. So the genetic code simply is a mathematical way of saying if you have 4 letters, let's just stick to blue. If you have 4 letters in the RNA, that's going to be A, C, G, and U. And at any one location, you can have any one of those 4 letters. And the word consists of 3 letters. How many possibilities are there, right? And we can use a mathematical formula, which says simply how many letters can occupy one space and how big is the space. So in this case, at any one location, there could be any one of these 4 letters. And then in this case, the word is 3 letters long. So we get 4 times 4 times 4. And that means mathematically, the maximum number of words we can have is going to be 64. Okay. There you go. Right, so 4 times 4 is 16, 16 times 4 is 64. So there's 64 possibilities. And if you look in that table, there will be 64 boxes. There's 4 in each row, and there's 4 rows. And that adds up to 64. So every single word that's possible mathematically is used by the cell, sometimes to mean the same thing, but in most cases to mean something unique. And the genetic code is simply a way of translating the codons. This is the codon here. So say it's C, C, C. So translating that codon, you look in the table, you look at the first base over here. So it's going to be a C. Right, so let's do that over here too. So the first base, let's put it in red. So the first base that we have over here is a C. So we look in the column with the C. The second base is a C. So we look in the top for another C, and they intercept right here. The 2 C's intercept right there. But we haven't finished yet. We then have to look for the third C, which is right there, just on that little tiny row. Then we look across, and there we find them, the 3 C's. So that word, CCC, in messenger RNA will be translated by something into which amino acid? Proline. Proline. And that's the way the genetic code works. Now, out of these 64 possibilities, there are some special ones. And there's 4 special ones. One of the 64 is going to be called the start codon. That means that's always going to be the first one to be used when making fresh polypeptide. And we look carefully, we can see AUG is the start codon. So in most living things, AUG will always be the first codon that a machine reads to start producing polypeptide, right? There's 3 other codons that are going to be the stop codons. So out of 64, there are going to be 3 codons that tell the system, when you get to the end of the polypeptide, no more, you're done. There's no more information that I want you to put into the polypeptide. And those are here. You can see them right there. There's 2 there, and there's 1 there, right? So they both start with the letter U, or sorry, all 3 of them start with the letter U, and one is UAA. The second one is UAG, and the third one is UGA. All right. So if you can remember these, that'd be good enough for us. So this is easy. AUG is August. August is the beginning of the academic year. So it's the beginning of the polypeptide. There you go, right? And that always brings in methionine. That always brings in methionine. You can use AUG inside the polypeptide, and then methionine will be put in there too. But the very first nucleotide is always going to be methionine. And it can be modified later. Once the polypeptide is finished, you can chop off the ends if you want to trim it down, and then the methionine will be lost. But it's always made first. So these are the rules by which nature works. All right. So this is the genetic code. If you look carefully, some of these like leucine, you can use 6 different words to mean leucine. So that's why these 64 possibilities that you see here, minus these 4, that gives you 60, right? So out of those 60, then some of them will be distributed amongst the 19 amino acids. So that means naturally some of these 19 amino acids will use more than one word. And that's what this indicates here, right? In fact, the vast majority of amino acids in that table use more than one word to mean the same thing. I'm going to look at that in a few moments. Let's just pop over to the next slide and understand this figure here. This is just terminology. Students must understand terminology in genetics. So we have the template strand, we have the coding strand. There's all these different things, right? So if you look carefully, the bottom strand, by convention, by rule, the bottom strand in a sequence can be assumed, unless you're told otherwise, the bottom strand in a sequence of DNA can be assumed to be the one that's physically used as a template by the RNA polymerase. I was going to say DNA, but it's the RNA polymerase, right? So if you have A on top and you have T below, then you have T on top and you have A below. This is base pairing, right? You have C on top, then you must have G below. So this represents one strand of DNA. This represents the other strand of DNA, right? Then the bottom strand will be red. And it has to follow the rules as to which way things are read and which way things are made. Remember the rule. Whenever you make nucleic acids, like for instance here, when the enzyme RNA polymerase reads the T at the beginning of the messenger RNA, or the RNA in this case, it will put in an A. And when it reads an A, it's going to put in a U. And when it reads the G, it's going to put in a C, right? So as the messenger RNA is built up, it's going to mimic the top strand. Yeah. So the top strand is called coding because it has the same information as the messenger RNA, or the RNA if we're looking at non-protein coding genes. So we can take that off. We don't have to put the M in front because it could be any type of RNA, right? So the rule is that all nucleic acids, DNA and RNA, are made backwards. So this applies to RNA of all types, not just some types. So DNA and RNA are made backwards. So that means 5, 4, 3. So this will be the 3' end. And when the messenger, or the RNA is finished, this will be the 3' end. That will be the 5' end. Because base pair takes place in anti-parallel direction, this must be the 3' end of the bottom strand of the DNA. And that somewhere over here will be the 5' end. And then likewise, because of base pairing and the anti-parallel nature of DNA, that will be the 5' end, and that will be the 3' end, right? So you can see the sequence here and the sequence here. So the coding strand is going to be identical to the RNA, except that all T's are replaced with U's. If you look carefully here and you read the sequence on the top strand, if I do it that way, you can see it. Doesn't matter if you go all the way across, every time you see a T, don't forget to replace that T with a U, right? And that is the only difference. So even though the bottom strand is used, because of this mirror image thing, the bottom strand will resemble the coding strand, which is not used by this particular G. All right, so those are simple rules. And then once the messenger RNA is translated, because now it has to be messenger RNA, right? Because we're going to translate it. When the messenger RNA is translated, then one of the AUGs, that's going to be somewhere located inside the messenger RNA. That AUG will be discovered by the system, and then a methionine will be put in, right? So you don't start copying from the very first nucleotide. You don't start reading the messenger RNA from that. You have a little bit of extra at the beginning, and you can see that here. the white boxes indicate where the start codon is, and every codon thereafter is going to be translated using the genetic code. And then the stop codon in this simple example is right there. And then the machine, the ribosome, will stop making further polypeptide after that point, right? So there's a lot of information in here. And this figure has some very important words. So look at this, the untranslated region. That simply means the part of the messenger RNA that is not converted into polypeptide, but is used to attach itself to the ribosome. Yes, the ribosome needs to attach to the messenger RNA. And that little bit at the front, which can be different in different genes, needs to be present to carry that information. Attach to me and then find the AUG. And then the ending is also important. The far side that's not converted into polypeptides, amino acids, is called the 3' untranslated region. So these are the most important regions that we'll consider in another chapter when it comes to regulating how much protein you're going to make and whether you can modify that protein or not, right? So you see the relationship. This figure here is very important. If you can explain this figure to somebody else in a lot of depth, then you're doing really well with the first half of this chapter. So let's move forward, otherwise we'll be stuck here all day, all right? So this is the conditions I was talking about earlier. So the genetic code is nearly universal. In mitochondria, we have some weirdness. Yes, AUG doesn't mean methionine. AUA stands for methionine. And there's a weird alternative amino acid called the 21st amino acid that we don't normally talk about in cell biology, but we do in this class. Selenocysteine, that's found in creatures, and including humans, and it can generate an alternative form of cysteine. And that's encoded by, in some cases, UGA. But how can the same word be used for two different functions? Well, those are the secrets of the cell, right? That we're beginning to understand. And you don't need to know that for this class. And then we have a 22nd amino acid, pyrrolysine, which is also a modified version of a stop codon. And that's normally found in archaea, which are prokaryotes. So you can see different branches of the tree of life are beginning to show signs of breakaway. Yes, that's amazing. Wonder what the world will be like in about 1 million years or 10 million years from now. So here's a summary of the genetic code with the exceptions, right? So the exceptions are given, and it's up to you to then remember some of these. Okay, please. All right, so let's move on and talk about polypeptides. So you should know from simple chemistry that you learned in a previous class, like Bio 2, that polypeptides are not branched. They don't have side branches like sugars do. They are simply a beginning, some amino acids in the middle, and then an ending. And because amino acids are put together in a very highly ordered mechanism, there's always a beginning. And the beginning of the polypeptide will have a nitrogen atom at the beginning. So that's called the N-terminus. And the very last amino acid to be put in the polypeptide will have a carbon atom at the end. So that's called the C-terminus. So just like with DNA, polypeptides have a defined orientation. And just like with DNA, which is made backwards, technically, if you look at the N and the C in the alphabet, polypeptides are made backwards too, starting at the N-terminus and finishing off at the C-terminus, wherever that may be, right? And the other thing is, you should have learned by now that these strong bonds that are holding the amino acids together, these are covalent bonds, very strong. And they have a special name. They're called peptide bonds, right? So there's a peptide bond being formed, and now the peptide bond is complete. The basic structure of the backbone is NCC. So that will be one amino acid. That would be the next amino acid. There's your third amino acid. and then there's your fourth amino acid. And then the next one will be attached here, and the process will continue for as long as this polypeptide has been coded correctly by the gene. So the gene decides when to start, what the internal amino acids are, and when to stop. Right? In most cases, there's some exceptions to the gene deciding. Sometimes environmental conditions can decide too. All right, so that's how polypeptides are structurally orientated. So you need to remember that. Here we can see the relationship between messenger RNA and the corresponding polypeptides. So we always read from left to right, and we always start with the 5' end of the messenger RNA. That makes the N-terminus of the polypeptide. So both of them, either the far end of the numbering system, 5 rather than 3, or the far end of the alphabet, N rather than C. And the side chains, of course, give properties to that local area of that polypeptide and its interaction with the environment, right? So the primary sequence is the order in which the amino acids are put together by the ribosome. The secondary structure is how, once that part is made, how it begins to fold on itself. So if my hand is made first, and my elbow is made second, and my upper arm is made third, then my hand will fold into a shape before my arm has even finished being produced by the ribosome. So that, if it takes up a spiral structure, like a helix, that's going to be called an alpha helix. So there's going to be a secondary structure. My arm may fold into a zigzag. That'll be another type of secondary structure. So those are the two most common types of secondary structure, but there's many more. And the secondary structure is basically getting its instructions from the primary sequence, right? And then once the whole arm has been made, it will then fold up into a final structure where the hand folds onto the elbow or the hand folds onto the upper arm, and then you get a final shape. And 2/3 of your proteins, they are done. They're ready to do their job. So 2/3 of the polypeptides consist of, 2/3 of the proteins inside us are finished, and they consist of a single polypeptide. The remaining 1/3 of our proteins, they are complex. They are made of lots of subunits, i.e. more than one polypeptide coming together to make a larger protein, like hemoglobin, which consists of 4 subunits. So that's going to be represented about 1/3 of your protein complement, right? So that's indicated, the secondary structure is indicated here in this panel. This is secondary structure. This is primary structure. This will be tertiary structure. So we just zoomed in on that. There's an alpha helix and there's a beta sheet. And then these are called unstructured domains. They have flexibility compared to the other parts. And then for most proteins, they're done. This is it. This is how they go to work. But some of them, they may have, 1/3 of them may have, 1/3 of them do have a quaternary structure, right? So here we have 2 polypeptides, the polypeptide below and the polypeptide on top. Sometimes they're made of the same 2 subunits coming together, so 2 right hands coming together. Sometimes it could be a right hand and a left hand coming together, or it could be a right hand and an elbow somewhere else coming together, right? So we have variation. So knowing your protein structure is very important. And finally, we know that the 20 common amino acids, they fall into roughly equal buckets. Each bucket is either going to be water loving or water repelling, right? Disliking. And that's going to then allow different proteins to take up different structures in 3-dimensional space. That's going to be very important when we look at mutations coming up. If you change the location or the identity of one of these amino acids, then that can lead to a misfolding of the secondary structure, which can have a knock-on effect on the tertiary structure, which can have a knock-on effect if you have quaternary structure. And that can then have a phenotypic effect on the organism itself. So it can be that severe, okay? And these are the hydrophobic amino acids. You count them, there's 2, 4 on the top row, 6, 8, 10, 11. And on the previous slide, 2, 4, 6, 7, 8, 9, 10, 11, right? So some of these have similar properties. Okay. So what we're going to do now is have a little experimental look at something. The reason is because Gobind Khurana was a student at the University of Liverpool, where I got my genetics degree, and I made it a point in the lab to sit in the same chair that he used to sit on. That's what the professor said anyway. So I wanted to be inspired, right? He got the Nobel Prize and I haven't. So there's something wrong somewhere, okay? So what did these scientists do? He was one of them. He wasn't the only one. There's about 6 scientists that worked on this. Well, in those days, in the 1960s, we wanted to learn how the secrets carried in the DNA were being coded into the different amino acids. We wanted to learn the genetic code. And compared to today, the technology then was simpler. The tools were more limited, but the scientists were just as clever. And they set up these artificial systems where they could feed the ribosomes a messenger RNA that they made using their chemistry, and then figure out what product was made by the ribosome, right? So let me give you an example. If you fed the ribosome a string of UCs, so UC, 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 then you waited to see what was produced in the test tube by the ribosome produced just a string of serines and leucines. That's the only thing it wanted, okay? You did the same experiment using other combinations, right, of synthetic RNA. They were able to then produce alternatives. To cut the story short, what they discovered through their ingenious experiments was that if you had a set of words that were 3 letters each, they produced a variety, sorry about that, they produced a variety of amino acids in the right order, as you would expect in a living system. When you had 2 letters only, it didn't. When you had 4 letters, it didn't. So that narrowed everything down to the words in genetics being 3 letters each, called now the codons, right? So CCC, as we said before, codes for proline, and that is exactly what they got in the test tube when they put CCC into the system. All right. So how does that work? Well, the magic bullet in this case, the magic recipe is the ribosome. And the ribosome, we'll talk about the structure in a few moments. The ribosome is where the magic happens. The ribosome itself is made of proteins and ribosomal RNA, yes, but it can't work on its own. It needs information. And where's the information coming from? Messenger RNA. What else does it need? It needs transfer RNA to bring in the right amino acid so that it puts the right amino acid in. So the linchpin in this whole system, the most important piece would be the transfer RNA. Because if the transfer RNA is trusted with the wrong amino acid, the system doesn't check any further. It'll put in whatever the transfer RNA gives it. So if the system is believing that the transfer RNA will carry and bring the right amino acid when it reads the right codon. Now, we're going to look at the tRNA in a few seconds, right? But it's important to realize that this is a biological machine, like a sewing machine. What does the sewing machine do? It reads the pattern on the messenger RNA. It uses thread that's brought to it by the tRNA, and then it makes a product called the polypeptide. And that is the ingenious nature of this translation system. Okay. The tRNA, represented as a pink ribbon in this textbook, is in fact a pure RNA molecule. It's a tRNA, transfer RNA. But the tip of the tRNA is loaded, it's attached covalently to a defined amino acid. So this particular population of tRNAs that are made by a particular gene they will always be loaded with this amino acid, phenylalanine. Another gene makes this tRNA. This tRNA will have a different sequence, and it will also be loaded with an amino acid, but not phenylalanine. This sequence is different, therefore it'll be loaded every time under normal circumstances, which is almost always with proline. So there'll be another 18 more of these tRNAs, right? The bottom end of the tRNA has a complementary set of bases. Remember, this is RNA, it's not protein, it's RNA. That has a complementary set of bases, which are complementary to the sequence of the messenger RNA. So this is the codon, therefore that is called the anticodon. That's what an anticodon is, right? Anticodon simply fits by base pairing with the codon. And you can see here, if the codon says UUC, then the anticodon will be AAG, and that will bring in phenylalanine. If the codon says CCG, then the anticodon will be, well, how do you read this? Do you read it GGC or do you read it CGG? That's what we're going to learn, right? That's what we're going to learn. But you can see the messenger RNA is always going to be orientated at the left side with the 5' end, on the right side with the 3' end by convention. Here's a picture, a magnified picture of what a tRNA looks like. It's a RNA molecule made by the process of transcription from its own gene. But it's also modified once it's finished. So the tRNA is then modified by enzymes, which are present inside the nucleus and in the cytoplasm, and they modify this. So some of these bases are going to be modified, right? So there's a modified base here, there's a modified base there, there's another one there, and there's your key. Modified simply means chemically altered once they're put in place. And the tRNA will also have internal base pairing, so it can form these stem loop structures, these hairpins, of which there are 4, and the anticodon is always going to be down here, almost halfway into the molecule. That's going to be the anticodon. And this part is very important because this part is used by a different enzyme system in the cytoplasm, which attaches the right amino acid, as we'll see on the next couple of slides. So these tiny little molecules are the linchpin to life. If they got things wrong, there's no quality control check after that. They have to be trusted, all right? So how do you put the right amino acid on the right tRNA? Well, like I said, there are enzymes that will fit the tRNA and the amino acid simultaneously based on both their shapes. And if they both fit properly, then they'll be bonded together with a covalent bond. What are those enzymes called? Well, they're called synthetases, right? Synthetases. What do they synthesize? Amino acyl tRNA synthetases, okay? And there's going to be one of these for each type of transfer RNA and each type of amino acid. And they use a lot of energy, and that is made by the cell, ATP, to do their jobs, okay? And you can see in figure 1511 how there's 2 active sites on the surface of these enzymes, and both of them have to fit their substrates perfectly. And if they do, then energy is used from ATP to bond one, the amino acid, to the other, the tRNA. And once that has happened, that product is released, and that becomes a pool of tRNA ready to be used by ribosomes, and the enzyme is ready to do the recharging with the next substrate. So these enzymes are working full time, all the time, inside the cytoplasm of your cells, right? So are there any complications? Well, if you remember back here, I told you that there were only 19 amino acids left after you take out the methionine, and that there's 60 possible codons. How do you account for this overlap, where one amino acid can be coded for by multiple codons? Well, that's where this comes in. We have something called the Wobble Rule, right? What it says is that of these 3 positions, this is the 5' end, right, of the messenger RNA. Let's put that here. This is the 5' end of the messenger RNA. and this is the 3' end, the first word, this word here, the most important thing is that you get the first position right. And you get normally the second position right. The third position, it's a bit flexible. It's a bit flexible. You could have an A here, a C, a G, a U. So for some words in this relationship, the third position is less important than the first two. And that's what the Wobble Rule says, right? So when you compare the codon table, which is back here, let's go back quickly, because you should do this, because this is reinforcement of that principle, right? So let's go back to this table. Here it comes. Oops, didn't know it's so far away. Here we are, okay? So you can see here, let's just consider the top two, right? This is phenylalanine. So if you look in the table, there should be nowhere else where there's phenylalanine except these two codons. And the order is same as this, right? The first letter in this codon will be the 5' end of the messenger RNA. So there's going to be a U. The second one is going to be a U. But look at the third one. The third one can be either a U or a C, just like we were saying here, right? So that's going to be forgiving. So U and C are interchangeable. So it's going to bring in the same amino acid. So that's where the wobbler happens, right? If you look at another one, let's go and look at serine. This is serine, right? Is there anywhere else that there's serine? So you have to look around. There's serine here too, right? But let's just look in this block. So for these 4 words, look at the first letter. It's always going to be U. The second letter is always going to be C. The third letter can be any one of the 4. So there's your wobble. So that's what the wobble hypothesis says, is that we know how nature has accommodated this difference in the number of amino acids and the number of codons that are possible, the 64. So there's your wobble, okay? And if you have any concerns about the wobble rules, you can look it up on the internet. There's a nice video or 2 out there, right? And that's very important. What is an iso acceptor tRNAs? Well, iso means same. So those 2 tRNAs that carry a different anticodon, and they differ at the third position between each other, they will carry the same amino acid. They will carry the same amino acid, right? So that's how the whole system has worked out a agreement amongst themselves, so that biochemistry can do its job inside your cells. All right. Okay, so wobble rule is always hard to explain, but it has to click in, right? So if you have the word Y, WHY, you could replace the Y with an I, WHI. And it still means Y, but it's spelled differently. Did you get the idea? Yes. Hopefully you do, all right? So what did we find? Well, there's another deeper rule, right? If the third base, i.e. this one here, if the third base in a codon is an A, the anticodon can be any combination of these real and modified bases. Oh my God. You don't have to learn them. All you need to know is that there's a lot of variation on the third base, right? And mostly the third base. Sometimes the second base can vary, but not ever the first base. So if the third base happens to be a U, that is this one here again, then it can be any one of these plus these modified bases. If the third base, the third letter in the codon is a G, then it can be any one of these, and then this modified base. And if the third letter in the codon is a C, it can only be one of these and nothing else. So you can see C can be substituted by an A or a G. Okay. So enough said. Let's move on and look at the way the machine physically is constructed, the ribosome. So we're going to go to the next slide and look at the ribosome. One thing to realize is that all living things have ribosomes. In fact, in bacteria, they have one type of ribosome, right? In eukaryotes, we have one type of ribosome in the mitochondria, and the chloroplasts. Yeah, if you have chloroplasts, your plant cell, you'll have a certain type of mitochondria. Sorry, if you have a plant, you'll have mitochondria and chloroplasts, and they will contain a population of ribosomes. But you also have ribosomes in your cytosol, and these ribosomes are generally a bit larger, right? So these are equivalent because of the history of life, the endosymbiosis theory, how mitochondria used to be ancient bacteria, how chloroplasts also were a different class of ancient bacteria that had their own ribosomes. So that branch of the tree of life developed its own ribosome, its own sewing machine. And then we have our cells with our own sewing machines, which are a bit bigger, more industrial than theirs. But they're both, the two types of sewing machine, they produce the same product, which is polypeptides, right? So the end product is the same, but the construction of the machines is slightly different, right? So these are prokaryotes, and these are eukaryotes. Eukaryotes, E, not K, right? So what we're looking at here is the overall architecture of a ribosome. So ribosomes, when they're not being used, when the machine is idle, is not being used, the ribosomes are deconstructed into two halves, the small subunit and the large subunit. But when they're needed to produce polypeptide, the small subunit and the large subunit are brought together with other players, such as messenger RNA and tRNA. So we're going to learn that very quickly. If you know this, you can skip this part here. Now what you see there is the same thing we see on the second slide, right? So the top part of this is the same as this side, and the bottom part of this is the same as the bottom end here, right? So these down the bottom will be your eukaryotic ribosomes. And it simply says that when you do some type of experimental analysis on this, this one here is called the 40S. And S stands for Swedberg. Swedberg, he was a Swedish researcher who did these types of measurements. And this was called the 60S, right? 60 Swedbergs. Whereas this is the 30S, and this is the 50S, right? So when they're separated, that's the kind of density they have in a centrifuge. This too is a 30S, yes, and this too is a 50S, even though they're mitochondrial and chloroplast, right? So you can see where the evidence is coming in that they are more related to these than they are to us. Regardless, the important thing is that these are further constructed of parts. So let's just look at one of them. So this 30S, when you start taking it apart, it falls into two parts. The RNA on top, which is going to be the 16S RNA, oh my God. And then we're going to have 21 polypeptides. So you can see that here, right? So the 30S has 21 polypeptides, and then it has one ribosomal RNA. Likewise, this 50S is going to consist of 34 polypeptides, and it's going to consist of 2 ribosomal RNAs. Okay, good, times 2. And this is common to all prokaryotes. What about this large ribosome? Well, it too is constructed of 2 parts. It's constructed of RNA, that's going to be the 18S, and in this case, 33 proteins. And how about the large subunit? That's going to be composed of something similar. It's going to be composed of 3 RNAs, ribosomal RNAs, right? 3 of them, ribosomal, and 49 proteins. I'm going to have to look over there because you don't have to remember these numbers, but you have to look at the pattern, right? So small subunit has 1 RNA and a bunch of proteins, and the large subunit normally has 2 or more RNAs and even more proteins. And when you look at this, and the question then becomes, what does this look like? And the more you study the endosymbiosis theory, the more you say that they look like these than they look like the other ribosomes inside our cells. All right. So all that information is coming from figure or table 50.4. Okay. Ribosomes are so good at doing their job that when one attaches to the right place on the messenger RNA and it moves down, doing its job, another one will attach behind it. That's a possibility. And when you get that kind of an anatomical arrangement inside the cell, and you can see it with the electron microscope, you get pictures like this. And here you can see you have a train of ribosomes going down this red messenger RNA. And as they go down, they start producing a polypeptide, the green lines, right? So these ones are just attaching. These ones are producing small polypeptides. And as they go further down, the polypeptide string gets longer and longer, and the longest strings will be over here. So what do you call the entire arrangement of these pieces? You call that a polyribosome, or polysome to be short, right? So polysomes are a more efficient way of translating the information in messenger RNA into polypeptides. And we do find them in both eukaryotes and prokaryotes. It depends on how important that gene is and how much product you need in a specified period of time, all right? Okay, so the final part, well, not really, the penultimate part of this chapter talks about the machine itself. How does it work? So it's like looking at a sewing machine and seeing, you know, the foot, the needle, the spindle, and everything else, right? The spool and the workings of a machine. Well, as far as we need to know, there are three parts to the ribosome, and they're called APE, or APE, or EPA, Environmental Protection Agency. It doesn't matter. The important thing is that the sites in all these ribosomes that you see on the right-hand side, they all have these sites. So they have the A site, I call that the acceptor site, the P site, which I call the polypeptide site, and then the E site, which I call the exit site, right? So the ribosome will be moving in this direction down the messenger RNA. So the ribosome will attach here somewhere and then move down the messenger RNA until it gets to the stop codon, right? So the ribosome is moving the messenger RNA through its body in that direction, right? So if this is the ribosome, the messenger RNA is passing like that, and the ribosome is moving that way. There are three phases to every ribosome doing its job. We call them, we call them initiation, the beginning, elongation, the continuous part, and then we have termination. So what you're looking at here is the initiation phase. Why? Because the messenger RNA, the first tRNA carrying methionine, and the large and the small subunit of the ribosome, they attach in this orientation. And then initiation is complete. The machine is now constructed and is ready to put in the first amino acid. Then elongation would be the next tRNA coming in and landing right here in the A site, and then matching itself to the codon. And then you have the second amino acid, which is part of elongation. All right. So elongation is a continuous process, and that will continue until you get to the stop codon, and the stop codon will be termination. So here we have the parts coming together. They form the initial machine, right? The initiation complex. Once the initiation complex is checked and is happy, it will then elongate. Elongation will continue, where new amino acids arrive here with their cargo of, new tRNA arrives here with its cargo of amino acids. And then the amino acids will be bonded together through peptide bonds. And the empty tRNA that has left behind its amino acid will then be ejected. And then that tRNA will go back to be recycled and recharged by the enzymes that are out there, right? This process will continue for as long as the messenger RNA is telling it to continue, until it reaches the stop codon. And the stop codon is in yellow here. And when it reaches the stop codon, there is no corresponding tRNA, there's no anticodon, but there are proteins that will be called in, and they can match the stop codon in terms of stereochemistry. they'll land in there. They're so bulky, they cause the ribosome to become unstable, then it falls apart. And then you have the polypeptide released. It will now start folding into its secondary structure and tertiary structure. And then the whole system is now ready to be either recycled, or you can use a new piece of messenger RNA, right? So you have to learn the steps, unfortunately, or fortunately, because it's a beautiful process. What other players are missing in that slide? Well, there's other proteins here that you don't see. What are they called? Well, in prokaryotes, they're called IF1, IF2, IF3, initiation factor 1, initiation factor 2, and initiation factor 3. And in bacteria, the first tRNA is called transfer RNA for methionine, but it's called F-methionine because it's called N-formyl methionine. It's just a modified version of the normal methionine, right? There's two types of methionine. And the start codon, as we said earlier, is going to be AUG, although there's now evidence that sometimes GUG and UUG can be used, right? But the first amino acid put in is always going to be methionine. And the same is true for eukaryotes, okay? But let's keep looking at bacteria. The person that discovered that the ribosome has a particular job to do, where to start things and where to stop things, that person or scientist, they were called Shine Delgado, right? I'm not sure if it was one scientist or two. I'm not sure. It could be Shine and Delgado or Shine Delgado. I'll have to look that up. Okay, so the way this works is that the small subunit, the 30S, this one here, it binds by itself to the messenger RNA first, right? And it looks for the Shine Delgado sequence. So it scans the messenger RNA looking forward and looking backwards, looking for a particular sequence, a consensus that it wants to find. Now in bacteria, this is very important. In bacteria, the messenger RNA and the small subunit, they come together randomly. So sometimes the small subunit will attach to the 5' end, the beginning of the messenger RNA. Sometimes it'll attach to the middle. Sometimes it'll attach to the end. It doesn't matter. When it attaches, it scans up and down, looking for the nearest Shine Delgado sequence, right? And when it finds it, it stabilizes itself. And then it knows, okay, I need to go to the right, because that's the way I should be translating this messenger RNA, right? Yeah, and you can see the 16S RNA, which is part of this, there it is. So that 16S RNA, because it's RNA, it carries bases. And those bases match up to the messenger RNA bases. Oh my God, you get base pairing. And that's how it becomes stabilized. So the heart of the machine called the ribosome is a small piece of RNA. Without that RNA, life as we know it would not be possible, because no ribosome would work. There would be no proteins on the planet. There would be no life. This relationship that you see down here on this slide is paramount. Only then is the ribosome primed to start making polypeptides. Okay, very, very important. And you can see hydrogen bonds are represented by dots, 2 hydrogen bonds between an A and a U, 3 hydrogen bonds between a G and a C. And this area, yeah, it can be, you know, rich in GCs, but it doesn't have to be. The consensus says, well, that's fine. It's okay. It's an even. And then if you look downstream from the Shine Delgado sequence, you will find an AUG somewhere in that vicinity. It doesn't have to be 2 codons away. It could be in that vicinity, right? But it's going to be close by. And now everything is ready to start using the AUG to bring in the first amino acid. So that is very important, okay? And you can see it taking place in this figure here, and it carries on to the next slide. So you may want to somehow learn this in the order in which it's presented. So that first slide, this is the second slide. Now everything is being primed. So initiation is now complete because the large subunit has come in and it's made those 3 caverns, the A, P, E sites. Now the system is ready for elongation. and then elongation takes place, right? So that's priming. Now, translation in us, eukaryotes, very similar, except we have more proteins that help our initiation process. And these factors are called EI factors, right? Eukaryotic initiation factors. So you see EIF6, then you know that you're talking about ribosomes in eukaryotic cells. If you see just IF, then you have initiation taking place in prokaryotes, right? And again, the way that our messenger RNA attaches to our ribosome is slightly different. Remember, we have a cap on one end of our messenger RNA. And that cap is very important in attaching the ribosome. So our ribosomes will always attach to the fingernails of the messenger RNA, never in the middle or anywhere else. Whereas bacterial ones we can attach anywhere. Our ones will always attach here, then slide down until they find the first AUG. Then they'll start translating from that point onwards. In bacteria, they can attach multiple times. You could have multiple start sites in a single messenger RNA. We're going to cover that in the next chapter, right? So here's a summary of some of those factors. And there's also release factors that are found that are used with the stop codon to dismantle this translation machine, okay? And there's some summary here that you should find useful as the order in which things come together. So compare that to the prokaryotic figure that you saw earlier, and you should be in good stead to understand this, okay? So the elongation phase is more or less identical between the two, except the speed. Bacteria, they, for evolutionary reasons, can add per second the correct amino acid at about 15 to 20 per second. Wow. Whereas we're a bit slower. We can only add about 5 amino acids per second, right? That is slow, but still fast. You know, count to 5 in 1 second, it's really hard. Now count to 20 in 1 second. That's going to be almost impossible, right? So speed is very important. And bacteria can respond faster by making new proteins much faster than we can do. But that's not the only advantage they have. They have other advantages too. So here we're seeing elongation taking place. Yeah, you can see how the A site and the P site work in coordination, and the polypeptide is beginning to grow and grow and grow. All right. So termination uses release factors, and those are the most common in yellow stop codons that we find in living systems, right? And then finally, this is just telling you the terminology again. RF immediately tells me I'm looking at a prokaryotic system, and ERF means eukaryotic system. All right. The mechanics, which site is used, how it's made into a polypeptide, the numbering of the polypeptide, the N-terminus, the 5' end of the messenger RNA, all that is the same. All that is the same. There's no difference. Now, let me just give you a side. Because of those similarities, we can take genes from humans or any eukaryote and give it to a bacterial cell. And the bacterial cell, if the messenger RNA has no introns, and it's processed messenger RNA, right? It's the finished product. We can give that messenger RNA to a bacteria. The bacteria should be able to correctly translate the messenger RNA into a polypeptide that has the same sequence as our cells would make. Well, isn't that the basis of biotechnology? Yes, we've been doing that for decades now. We've taken genes from humans, like the growth factor. We give the messenger RNA or the gene itself without the introns to the bacteria, and they make huge quantities of growth factor and insulin and many other substances, right? So that's the basis of biotechnology. If bacteria were completely different to us, we wouldn't be able to do that. So that kind of perspective opens our eyes too, right? So here's a quick comparison. You can do that at your own leisure. This is a summary of everything we've learned so far in a single table. This one simply says, hey, these differences that we have, let's go back to this slide here, these differences that we have in our structure between these two, that is important because now we can use certain antibiotics that bind here, but they won't bind here. Or they bind there, but they won't bind here because of these structural differences. But what about this? What about the mitochondrial ribosomes? Won't they bind the antibiotic? Well, the good news is mitochondria have a membrane around them, a double membrane, and these antibiotics cannot penetrate the inner membrane of the mitochondria. Therefore, they can't get into the space where these look-alike ribosomes of mitochondria are found. So the antibiotic can't penetrate into the mitochondria or the chloroplast. Therefore, we're safe. And the drug companies have to test for that. Amazing, right? So there we are. A detailed, but still synopsis of chapter 15. I want to thank you. Do you have any questions? You know where to find me. Thank you so much. Bye-bye.